In this video, we will review DNA and gene expression. Let's start with DNA replication. So before a cell divides, we have to make copies of that genome, and so this occurs through DNA replication. There are 3 stages. The first thing you have to do is unzip those double strands so you can access the individual nucleotides to replicate them. This occurs through an enzyme called helicase. During elongation, we have an enzyme called DNA polymerase that then creates a complementary strand for each template from our original strand. That continues along the DNA until it reaches the end, and our third stage is termination, where that process ends. For most of this video, we'll look at the central dogma of molecular biology. This is how information in our genes becomes our proteins. We'll start with a few terms. So a gene is a sequence of DNA that encodes a functional product, and this is generally a protein. Our complete set of genes is our genome, and genetics is the study of genes. From our DNA, from our template, we then transcribe that code into an RNA, and this is the process of transcription. From our RNA, we then translate that message into a protein. The complete set of mRNA transcripts from DNA is called the transcriptome, where the complete set of proteins encoded by a genome is called the proteome. And the study of this is proteomics. So our DNA is divided into 23 pairs of chromosomes, 22 pairs of autosomes, and 1 pair of sex chromosomes. These are discrete bundles of a linear DNA helix that is then tightly coiled and packaged into what we call chromatin. So here you can see chromatin is supercoiled to make it more condensed. We have specialized proteins called histones that package our DNA. You can see they kind of wrap the DNA around to package them up. And histones serve an important role of controlling access to our DNA so that we may express or not express a particular gene. And these are called histone modifications. If you add an acetyl group to a histone, so histone acetylation, this will allow for a gene to be turned on by opening that chromatin fiber, so then our products of transcription, our enzymes can then start transcription. You can also add a methyl group to a histone through histone methylation, and this closes up that chromatin fiber and turns the genes off. So from our code of DNA, we transcribe this into RNA. This process occurs in the nucleus, and the product is a messenger RNA. That's the message of that gene. And so while DNA is our blueprint of all of our genes, the heavy work really occurs through our messenger RNA. So in transcription, we have an enzyme called RNA polymerase that binds with the transcription factor that will then start creating a complementary copy of that gene in messenger RNA form. But remember that our DNA includes both exons, which are areas that code for a gene, and introns, which are non-coding regions. And so when we first make that messenger mRNA transcript, it will include both exons and introns. So to get a messenger RNA that we can use to make a protein, we go through a process called splicing, where we edit regions of the messenger RNA. This is a lot like film splicing, where you might cut out certain areas of a film and then adhere it back together so you have one continuous picture when you play it. And that's what happens here. we might remove introns or other segments for a particular isoform of a protein that we might want, splice it back together, and then we have our final messenger RNA. After transcription, we move on to the process of translation. Here we want to build proteins using amino acids that are bonded through a peptide bond. We have specialized RNA called transfer RNA that has something called an anticodon. So a codon is a triplet of nucleotides that correspond to a specific amino acid. And so these transfer RNAs will bind to the messenger RNA by matching its anticodon with the codon. That then allows for the transfer of the attached amino acid on this transfer RNA to this growing polypeptide chain. And so along the messenger RNA, these transfer RNAs will continue to bond every 3 nucleotides, dropping off an amino acid that's joined by a peptide bond to this polypeptide chain. Eventually, when you get to the stop codon, when you get to the end, this process will terminate and you'll have a polypeptide chain that is now a protein that will then function in the body. So what really matters here is our gene expression. This is where we transform information coded in a gene to a vital gene product. So is this protein or end product produced? That is what gene expression is. And so we're concerned about the degree to which a gene or a particular group of genes are actually being transcribed. So someone might have that gene, but is that turned on or off? And so when we turn on a gene, we call that process induction. And when something is going to turn a gene off, we call that process repression. And the gatekeepers of gene expression are the transcription factors. They initiate and regulate transcription. Another important piece is epigenetics. This is when there are changes in gene expression caused by something that's not a change in your DNA sequence, so it's outside, like those histone modifications. We are born with an epigenetic imprint, and this imprint is maintained when our cells mitotically divide. For example, you have a heart cell and a liver cell. These have 2 very different functions, and so they're going to have very different proteins that are expressed. And so epigenetics will control what genes are turned on and off in a heart cell and what genes are turned off and on in our liver cell so that they can do their respective functions, even though they have the exact same genetic code. But environmental and lifestyle factors can also modify our epigenetics. And so this might include things like physical activity, diet, or chemical exposure. And so these can lead to changes in physiology and potentially could lead to cancer. There's some different types of epigenetic modification. We have those histone modifications that we talked about. You can also add methyl groups to the DNA, and this can then create a physical barrier so that that transcription factor and RNA polymerase cannot do its job. Or you can go a little more downstream where we have RNAs that prevent the creation of a protein from a messenger RNA. And so these RNAs will bind to that messenger RNA and not allow this polypeptide chain to be built. And so the takeaway here is that even though we have the same genes in all of our cells, they're all expressed differently, whether it's because of different cell types or because of hereditary or environmental factors. The epigenetics can control our gene expression. And then finally, we'll look briefly at patterns of inheritance. And so we want to think about some different terms. So you should be able to distinguish genotype from phenotype. So a genotype is a person's genetic material. They both have GEN. And so this is that DNA sequence and composition at a given locus or particular part of a chromosome. The phenotype is their physical characteristics, and so they both start with PH. And this is the appearance of a trait, like blue eyes, or the expression of a disease. Genes are not always expressed equally, so people could have the same genotype, but their phenotype could be mild or severe. And so this concept is called expressivity. And then not everyone with the gene might express that phenotype. So we call this concept penetrance. So let's say 75% of people have a gene where they have an extra ear. If 75% of people with that extra ear gene have an extra ear, then the penetrance is 75%. So how many people with that gene actually have that phenotype? And that's penetrance. And so we all have two copies of a particular gene because we have two chromosomes, pairs of chromosomes. And so these copies of a gene are called alleles. If on a particular gene you have the same allele on each chromosome, you're considered to be homozygous. But if you have two different types of allele of that same gene, you're considered heterozygous. We have different types of trait. So a dominant gene will occur when you have that trait, whether you have one copy or two copies of that gene. For example, brown eyes are said to be dominant, that if you have one blue eye and one brown eye copy, then you tend to have brown eyes. A recessive gene requires two copies of that trait. So if you are heterozygous for a recessive gene, so you only have one copy, but you need two copies for the phenotype, then you're considered a carrier. So this is our brief overview of DNA and gene expression.